Vampire algae killers' genetic diversity poses threat to biofuels. New DNA analysis has revealed surprising genetic diversity in a bacterium that poses a persistent threat to the algae biofuels industry. With the evocative name Vampirovibrio chlorellavorus, the predatory pest sucks out the contents of the algae cells, thus the vampire reference, and reduces a productive, thriving, green algae pond to a vat of rotting sludge. New DNA analysis has revealed surprising genetic diversity in a bacterium that poses a persistent threat to the algae biofuels industry. With the evocative name Vampirovibrio chlorellavorus, the predatory pest sucks out the contents of the algae cells, thus the vampire reference, and reduces a productive, thriving, green algae pond to a vat of rotting sludge. DNA sequences show what are likely different species, suggesting a much larger diversity in this family than we originally assumed, said Blake Hovde, a Los Alamos National Laboratory biologist. That means the treatment for one algae pest might not work for another, which can be a big problem for large-scale algae cultivation in the future. The research team sequenced 2 strains of Vampirovibrio from the same pond. The 2 samples, collected 1 year apart, came from an outdoor algae cultivation system in the Sonoran Desert of Arizona, run by University of Arizona collaborators Seth Steichen and Judith K. The team sequenced and analyzed the genomes to identify the genes in. Our genomic analyses identified several predicted genes that encode secreted proteins that are potentially involved in pathogenicity, and at least 3 apparently complete sets of virulence (vir) genes, Hovde said. Those genes are characteristic of bacteria that carry out cell invasion. With chlorella algae valued as a key source of harvestable biomass for biofuels and bioproducts, it is extremely useful to be able to enhance the fundamental understanding of interactions between a unique bacterial pathogen and its green algal host, Hovde noted. The results of this research have direct relevance to the success of large-scale commercial algal production projects underway to advance U.S. energy security, biofuels, and the production of aquaculture feedstocks and algal-based nutraceuticals. For future work, the team is following up with a project with the Joint Genome Institute to characterize 6 more pest genomes from the same family to see if the diversity of these organisms continues to expand or if the researchers can start